I will share my screen. Yes. Can you see my screen now?
Yes, we can.
Good. Right. So I will take you a little bit on a journey through my travels through developing of therapies for rare disorders, and my main specialty is RNA therapies and for late onset neurodegenerative disorders. But I will take you through it. So these are my disclaimers. If you do translational research, yeah, and you claim to develop therapies for patients, you work a lot, collaborate a lot with biotech and industry. So here are my disclaimers. So the learning objectives for this webinar is that you would be able to give a general overview of the different types of genetic therapies, because there are many of them, and RNA targeting therapies are only a small portion of them. You can outline the major working mechanisms of the RNA targeting therapies using antisense oligonucleotides. I will talk mainly about spinocerebellar ataxia type 3, so you should be able to summarize the main disease characteristics of that disease. And you can give some of the examples of the different RNA targeting therapies that might be applicable for SCA3. And then I think it's important to also be able to distinguish the differences between the traditional clinical trial design and what if we want to use these RNA targeting therapies also for very, very small groups of patients. So I will first introduce a little bit the research in my own two groups. I have two groups now. I will explain what RNA targeting therapies I would like to focus on and how you can apply these to rare diseases, but also to nano-rare diseases or like nearly unique variants that cause disease. And for that, I will also outline a little bit the European and the worldwide efforts that we need to apply these NFU RNA therapies, because these patients are spread throughout the world. So it's no point making these efforts national, because the chances are that if there is a second patient, that patient will probably not live in the same country. And I will use for most of them as an example SCA3 to kind of build these cases. So this is sort of the daily bread and butter of the research that I do in my research groups. So first there is a DNA variant, and you need to figure out what the disease mechanism is. Is it because there is a lack of protein? There is a toxic protein. If you figure out what the cause of the disease is, you can then sit behind your computer and you can come up with a possible therapeutic approach. You first test that in cellular models. For neurological diseases, we now use a lot induced pluripotent stem cell models. If you have a fairly good idea that your drug is actually working as you were thinking of behind the computer, you will need to file for intellectual property, but only if you want to continue through the normal clinical pipeline of working with a biotech company and having phase 1, 2, 3 clinical trials. Then you need to show in an animal model that your therapy is working, and then the companies will start doing more of the more routine PK/PD studies, safety toxicology studies, things like that. And only then can you file to start with the clinical trial. So in my research group, I work for most of the time on polyglutamine diseases. I will explain a little bit later what they are. And really in my group, we are working mainly on splice modulating antisense oligonucleotides. I will also explain a little bit later about what that is. And for those, we work together or have worked together with different companies to bring them from the research lab towards clinical application. But as I mentioned, you know, we can, there are a lot of, there are millions of people that have a very rare genetic disorder. So there are only a few people with the same disorder, but if you count them up, combined, there are millions of people in Europe living with these rare genetic disorders. And this type of RNA therapy development can also can be used for these unique patients. So we have started the Dutch Center of RNA Therapeutics, where with 3 university medical centers, we are now trying to apply this in the Netherlands. This is something that was started in the United States. The Netherlands is part of the European Union, so we have the EU, we have the EMA, we have European legislation, but it turns out that each country has their own national legislation that makes it very difficult to do the same thing, even in 2 different countries like Germany and the Netherlands. So this is why we have also started the One Mutation, One Medicine network within Europe, to align all of these efforts of these NF-few RNA targeting therapies within Europe. And we work closely with the NF1 collaborative that was mainly based in the States, where Millicin, the first NF1 RNA targeting therapy, was developed in the lab of Timothy Yu. And we also work closely together to kind of align all of the efforts worldwide. So what are we talking about? So this is back to basic biology. So your genetic code is part of the DNA. After transcription, first the pre-mRNA is made. Then the boxes here, they depict the exons. So those are the coding sequences. And then the introns are the non-coding sequences. And in the step that's called splicing, these non-coding sequences are removed and you end up with the messenger RNA. And this is then what is being translated into a protein. Now, what comes back throughout the presentation is the shape of these boxes, and those depict the reading frame. So here you can see that these boxes, they fit nicely together. That means the reading frame is intact. So that means a protein can be translated. But if for some reason this reading frame is disturbed and these exonic boxes don't fit together nicely anymore, this mRNA is usually degraded through nonsense-mediated decay, and that usually means that there is no protein being expressed. Just to give you, so you don't have to look at this slide at all of the details, but this is just an example from a review that we've written in 2023 of all the potential therapeutic options that are under development or are in clinical trial for Huntington's disease. And this is on all of these 3 different levels. So there are approaches that are being developed on DNA level. So you can think of CRISPR-Cas mediated therapies, but also zinc finger proteins. There are a lot of therapeutic approaches that focus on the RNA. So they can be small molecules, they can be gapmers that will break down the RNA. There can be these splice modulating antisense oligonucleotides, but it can also be RNA interference. So there are many, many ways that you can target RNA that is being translated from the disease-causing gene from the DNA. But also there are a lot of therapeutic modalities that are being developed on the protein level. So I just want to give you a little bit of an overview to make sure that you understand that the particular RNA targeting therapies that I'm talking about is just part of a whole scala of therapeutic options. So I think antisense oligonucleotides are short single-stranded forms of DNA or RNA that can be modified, and they bind to the RNA through Watson-Crick binding, and they can either break down the RNA, and that is usually an antisense oligonucleotide, or an ASO as we abbreviate it. So this ASO can bind to the RNA, and this gapmer induces RNase H recruitment. It breaks down the RNA, so you do not have any protein that is being produced. However, you can also have an ASO that is slightly differently chemically modified, and that can bind to the RNA, and especially the pre-mRNA. And then it can interfere with this process that is called splicing. And then this makes it a very versatile molecule because there are many different mechanisms that this ASO can induce. So you have a variety of tools that you can use to develop your therapy. So I will go into a few of these mechanisms in my next slides. So what if there is a DNA variant that disrupts this reading frame? So I talked a little bit about the shape of these exonic boxes. If they fit nicely together, the reading frame is intact. But what if there is a DNA variant that disrupts this reading frame? And so when the pre-mRNA is spliced, these boxes don't fit together, so there is no protein expressed. And then what you can do is you can design an ASO that hides the splicing signals within this exon, so the splicing machinery doesn't recognize this as an exon anymore. It is spliced out, but because this exonic box now fits into this exonic box, you get a slightly truncated mRNA, but the reading frame is actually restored. So the result of this is that you can get a truncated but partially functional protein. Another way that you can use these ASOs, if there is a DNA variant that causes a toxic gain of function in the resulting protein. So again, what you can do, you can design a splice switching ASO that hides this exon from the splicing machinery, and then you get a truncated protein, but that misses the toxic gain of function part of this protein. So you will get a non-toxic protein that is slightly shorter, and hopefully it will have also retained most of its wild-type functions. And another variant the ASO strategy is very suitable for is if there is a DNA variant that is deep within the intron. And sometimes these deep intronic variants, they cause that part of the intron is included in the mRNA. And we call that a cryptic exon. Usually those are out of frame. And again, out of frame is usually means that there is no protein that is being expressed. So if you can then design an antisense oligonucleotide that hides this DNA variant deep in the intron, and you can show that normal splicing is restored, and then usually your normal protein expression is also restored. And actually, we really do feel that this type of deep intronic variant is really underreported because most of the diagnostic pipelines, they use whole exome sequencing, and we really quite often miss these deep intronic variants. So I think we are looking for the right thing, but maybe we should look both ways. We should not only look in the exons, but we should always also look in the introns. All right. So, you know of your DNA variant, you've been designing an ASO strategy behind your computer. You've got your lovely cell models, but how are you going to develop this into the first in-human studies? And actually doing that for a rare disorder is difficult enough, but if you are going to do this for a nano-rare disorder, that makes it even more difficult. So I'm going to give you a few examples. One that is for a rare disorder. So that is a clinical study that combines 3 different neurodegenerative diseases into one. So you kind of increase your study population. The second example that I want to give is an example for spinocerebellar ataxia type 3, where we use where we're developing an ASO program in the US under the Right to Try Act. And then I will go into details about the European N-of-1 pipeline, so developing an ASO for one particular patient, and how we are trying to implement this into Europe. So first, a short introduction on spinocerebellar ataxia type 3. So that is an autosomal dominant disorder. The prevalence is around 1 in 100,000. It's an adult onset neurodegenerative disease, and the main symptoms include ataxia, distal muscular atrophy, and paralysis. And the cause is a CAG repeat expansion in the ataxin-3 gene, and there is a threshold. If the CAG repeat expands over this threshold, you will develop the symptoms. And a CAG codon encodes for the glutamine amino acid. So a CAG expansion also means that in the protein there is an expansion of a stretch of glutamine. So we call that a polyQ expansion. There are actually, SCA3 is one of 9 of these polyglutamine disorders. So there are 8 more autosomal dominant disorders that are all caused by a CAG repeat expansion in the coding region of a gene. So for each of these 9 diseases, it's a different gene. And the most prevalent of these polyQ diseases is Huntington's disease. The next prevalent one is SCA3. And the other ones are even more rare. And the example that I'm going to talk to you about next is about SCA3, SCA1, and Huntington's disease. So here, this is a development here in Europe. So I'm going to go a little bit from Europe to the States and back to Europe again. And why is that important? Because the legislation is quite different between the US and between Europe. And in a way, that is, it makes it complicated. So for the first combined study that I'm going to show about HD, SCA3, and SCA1, the group is big enough to have a traditional drug approval process. So you have your target validation, your compound screening, your lead optimization, your preclinical tests. So this is all done, takes about 4.5 years. Then if you can collect all of your results, you will go in your phase 1 to 3 clinical studies. Then you go for approval to launch, and then there are post-marketing studies. So this is a drug, Vico has popped behind my agarose gel. But this is a study that has been performed by Vico Therapeutics in Leiden in the Netherlands. And actually, in my lab, we did the first, we published the first study together with people from, well, at that stage, there was still Prosensa Therapeutics. But so the mechanism of this ASO is actually quite interesting. So it's an antisense oligonucleotide that is composed of 7 CUGs in a row. So it really binds to the CAG repeat. And the idea is that in an expanded CAG repeat, more of the antisense oligonucleotide binds. So when the RNA gets to the ribosome, it can't get translated into a protein. So wild type RNA only has a few oligonucleotides, so the ribosomes can push them off, but the expanded repeat is being blocked from translation. And so I'm showing you the example for SCA3. So this is in a patient fibroblast cell line. So where you can see the mutant and the wild type expressed. And when we add lower and higher doses of this CUG7 ASO, you can see that most or all of the expanded ataxin-3 is no longer visible, but the wild type slightly reduced, but it's still there. And why is this important? So this is an allele-specific approach. This is important because we know all of these polyQ proteins, they have really important wild-type functions in the brain. And there is an ongoing discussion of how much of these toxic gain-of-function proteins you can lower to also get into trouble because you lose too much of the wild-type function of this protein. So that is why for a lot of these diseases, if we would have an allele-specific or an allele-preferential approach, that would mean you would mostly reduce the toxic gain-of-function protein, but you will leave the wild-type protein mostly intact. So this is now actually, so this CAG repeat, this ASO is applicable to all of the CAG repeat expansion disorders. And there's already a lot of preclinical work out there. So if you're interested in the specifics, you know, we can You can look it up, or we can discuss it a little bit later. But at the moment, you know, the preclinical work has been done by Vico Therapeutics, and they've just started a phase 1 to a clinical trial. So the participants, they get a monthly dose of this ASO, which is called VO659, total of 4 doses via intrathecal injection. So for most of the clinical studies at the moment, intrathecal injection is the preferred route of administration. And there's a follow-up phase of 6 months with different dosing levels. And each participant will receive only 1 dose level. So this is actually the first clinical study that includes these different poly-Q diseases. So this is one way of getting around the problem of having just such a small patient population to be able to still fit into this traditional clinical trial design. A minimum of 13 hospitals in 8 European countries will participate, and SCA1 and HD patients will be included from the 3rd dose level, and the study started with SCA3 patients. So the question is really, how low can we go? How small can our group of patients become to still fit in this traditional drug development pipeline? But really the question is, what will we do with these patients that are so rare that they will never fit this clinical drug development pipeline? And so now we move across the Atlantic Ocean back to the United States again. And we are just now going to talk about spinocerebellar ataxia type 3. So in the US, there is actually the Right to Try Act, and that is one way for patients who have been diagnosed with life-threatening diseases or conditions that have tried all approved treatment options, but who are not able to participate in any clinical trial, or there might not be a clinical trial in that country. And that will still give them access to investigational treatment developments. So for that, we work together with Cure Rare Disease. So this is really a non-for-profit organization that is really trying to push the boundaries of developing more appropriate pipelines to get treatments to these very small groups of patients faster, so they're not forgotten. So for that, we are using an ASO that is developed in my group. So again, so this is the ataxin-3 protein, and it's an important protein that has important functions in the proteasomal protein degradation. It cleaves off ubiquitin chains, and ubiquitin chains are markers that will mark proteins that need to be degraded by the proteasomal system. It has many protein interactors, and it has important transcriptional activity, and we think it has really essential protein function. So again, just downregulating it would probably not be a good therapeutic option. So here is the pre-mRNA, and again, you can see these boxes that depict the reading frame of the ataxin-3 mRNA, and you can see that the CAG repeat that is expanded in SCA3 is located in exon 10 of the transcript. So what my group has done is they've developed an antisense oligonucleotide that recognizes exon 10. So exon 10 is removed from the mRNA, and this was an approach that that in principle shouldn't work, because exon 10 is an outer frame exon. Excuse me. So if you remove this exon, the protein is out of the RNA is out of frame, and it should be degraded. However, right at the start of exon 11, we can see that there is a premature stop codon. So actually, this results in a prematurely truncated protein that lacks the poly-Q repeat, but it still actually has most of its functional domains. And we've shown in a few studies that most of its wild-type function that ataxin-3 has is actually retained. So this is also a way of maintaining wild-type function and only reducing the toxic gain of function. So we've tested it now twice in mouse models. So those are mice that express the human mutant ataxin-3. And you can see in the wild type mice, there's only wild type ataxin-3. In the mutant mice, you have the mutant protein. And when we add our antisense oligonucleotide, you can see that there is a truncated ataxin-3 protein. And so we always inject these antisense oligonucleotides in the ventricles of the mice. And you can see that both in the brainstem, which is further removed from the injection site, as in the cortex, we sort of get a decent protein modification throughout the mouse brain. One of the hallmarks of all of these polyglutamine disorders is protein aggregation in the brain. And here we did a filter trap assay where you can see that there is no insoluble ataxin-3 in wild type mice. The SCA3 mice that were treated with PBS, they show insoluble ataxin-3 protein. This is scrambled ASO that we have injected. But when our SCA3 ASO was injected, you can really see that there is a reduction in the insoluble protein in these mice brain. And as for behavioral studies, we've done several behavioral studies, but this was the most striking one. So this is the beam walk test, where you let the mice walk across a small beam, and you measure how long it takes to walk across this beam. You can see it in 8 weeks. There is no difference between the transgenic mice in purple and the wild type mice. But you can clearly see that after 18 weeks, the untreated transgenic mice and the untreated wild type mice show a clear phenotype. So the transgenic mice take longer to cross the beam walk. And then when you treat them with the ASO in the lighter bars, you can see that they can cross the beam faster than their untreated transgenic mice. So we're taking this program further so this goes through the investigational new drug filing through the FDA. So we still need to do some larger animal models for safety studies and do some dose finding studies. But yeah, this is a program that we are developing that is still ongoing. So an IND filing is a very elaborate process. So you first need to consult. If it falls under the IND, then you need to prepare and prepare a submission. The FDA will come back with comments. You have to incorporate these. And there is a whole procedure to follow during the trial, because it's still considered a trial. And in the end of the trial, you can determine whether your drug was successful or not. So this is all part of the IND document that you have to file for the FDA, and it is still quite conservative. They've made a white paper and to outline the things that they want to see if you develop a drug for a very small group of patients. But it is still quite an elaborate filing document. Then in 2019, the group of Timothy Yu actually custom designed an RNA targeting therapy for a DNA variant that was at that stage only known to be found in one girl in Milan. And this is her name. So the drug that he developed through this IND process at FDA was called Milasen. It was named after Mila. A few have followed. There are actually more now. They're all done in the States. But this is Ipek, who had ataxia telangiectasia. Her ASO is called Atipexin. Valeria, her ASO is called Valerius. And then JC, with her ASO, is called JC-Fusen. And so the IND filing for these N of ones is actually slightly smaller, a little bit more tailored towards developing something for one person, but it is still quite an elaborate document. However, we live in Europe, we do not live in the States. And when we started the Dutch Center of RNA Therapeutics in 2020, we started asking around to see how we could apply this NF1 and a few RNA targeting therapies in Europe. And we actually have not developed within Europe, started all the way from getting a DNA variant, developing the ASO and treating a patient. However, what we have done is we got the ASO that targets the mutation from EPEC or atipexin, because there was another patient that we identified in Germany that was amenable to the same ASO. So I'm showing you the first evidence of actually applying this NF1 NFU in Europe. So ataxia telangiectasia it's a rare inherited childhood neurological disorders, and it usually begins quite early on, and it's quite rapidly progressive. And it's caused by very many different mutations in the ATM gene. And those can be exonic, intronic mutations, missense, truncating variants. The neurological symptoms usually include unsteady walking and lack of balance, lack of coordination, slurred speech, difficulty swallowing, unintentional movements, and difficulty coordinating eye movement. So ATM is a protein kinase, and the most documented function of ATM is mobilizing and regulating the cellular response to DNA double-strand breaks. So the DNA variant from EPEC that was also found in this a little boy in Tübingen actually is an exonic variant that causes part of exon 53 to be spliced out, and the remaining mRNA is then out of frame. So there's a loss of protein expression. And the ASO was designed to cover this mutation, and then normal splicing was restored. So because the atipexin was designed and filed through an IND filing with the FDA, when this boy was discovered in Germany, we were not allowed to just get the ASO from Timothy Yu's group and apply it here, because the FDA wanted to make sure that the ASO was safe and not harmful when this boy was treated with the ASO. So first, the whole family had to fly to Boston, and that is where the treatment was initiated, and the dose escalation phase was done. And once the treatment was continued, the whole family could move back to Tübingen, and now it's being treated in Germany. So we are ongoing, and we're collecting all of the data, but also acquiring a lot of first-hand experience with applying these NFU ASO treatments within Europe. However, the legislation in Europe is quite different for these NF1 NFU trials than it is in the States. And actually, it's a chemical molecule, an antisense oligonucleotide, so it doesn't fall under the ATMP regulations. It's not an unlicensed, we cannot fall under the unlicensed access options for groups of patients. So for single patients, you either have hospital exemption, but that's only for ATMPs. So ASOs actually fall under the named patient use. So the named patient use does not have any regulatory approval. That's not necessary. That sounds ideal, but it really means that all the local medical ethical committees have to approve whether an ASO was developed and is safe enough and is effective enough to actually start treating a patient. So that really puts a lot of burden on all the local ethical committees to make this judgment call. Also, the hospital pharmacy is responsible for preparation of the GMP compliant or GMP-like compound. So also there, it's national regulations or institutional regulations that will determine it. And of course, we're worried about the pricings, because if we can do this for one patient, but every drug will cost then 3 million euro, I think we can stop this now, because that will never become something that will be affordable or will be reimbursed by the healthcare providers. So we'd like to set up all of these preclinical processes as much as possible in an academic setting that is still GLP compliant or GMP-like. And we already know that there are differences between the different European countries. So we like to coordinate from the start to also align all of the procedures that we're setting up so they can be applied throughout Europe. So what we're really trying to set up is going from a traditional drug approval process towards a patient-specific N-of-1 treatment. So there is usually no natural history data available from these patients, or it's so rare that the variability from one patient to another is already not enough to get a coherent natural history data for this particular disease. So what you really need to do is you need to use the disease progression, a unique disease progression profile of your particular patient. So from the moment the DNA variant was determined, and it was determined to be amenable to one of those ASO mechanisms that I've just shown you, from that moment on, you will have to start recording the course of the disease in your particular patient. So you will get sort of a run-in period where you will determine the patient's own natural history, that from the moment your ASO has been developed, you can then see if the progression deviates from the trajectory that you could predict from the run-in phase. So what we do in the One Mutation, One Medicine network is that we're really trying to standardize, is the variant amenable for one of those splice switching ASOs or GAPMER ASOs approaches. So we have case dossiers where everyone who has an interesting patient can fill out the patient details and the variant details. If the 1M1M core group decides that this is a patient that is amenable, so not too far progressed in the disease, or some patients have a variant where all of the damage is already done by birth, so there is not much function that you can still restore once you found the mutation. So the variant is assessed, the patient is assessed, then we go to a gene group meeting where we get more of the clinical history from the patient, all of the information that is available on protein function, RNA levels, protein levels, maybe an ASO was already developed. We will all discuss this in the gene group meeting. So that will be the clinician that has seen the patient, but we also ask for worldwide experts to join this gene group so we can all look at this very rare patient and the clinical profile. We also get domain experts and ASO biologists to see if the ASO approach is amenable. And then when all of this is assessed, we will go ahead and prepare for clinical treatment. And the treatment board is really the last expert meeting that we have that will have the final say if we will go ahead and actually proceed with treating the patients with the ASO was developed. So far we've had 2 or 3 treatment board meetings, but we've never given the green light because there was always some concern that we decided it was not really worth proceeding yet. But hopefully in the near future, we will have a positive outcome. So one of the things we're developing, because it doesn't really fall under the auspices of the AMA, they are really willing to help us setting up protocols, standardized protocols that we can then use to support the local ethical committees to decide whether the preclinical work and the clinical preparations are sufficient. So PRIME is a priority medicines program where AMA will help you to set up all of these protocols. And we've selected ataxia telangiectasia as the disease that we know most about within Europe, where we have hands-on experience with an N-of-1 ASO that we are applying to a patient. And they're helping us within this program to standardize the preclinical and clinical protocols that we can then hopefully distribute throughout Europe. And I think this is the group of people that I would like to thank. So it's my own research group, the DCRT, 1M1M, and the N-of-1 collaborative, and that was it.
Thank you very much, Willeke, for this nice explanation. And it's not only about biology, it's a lot about regulations also.
It's just, yeah. So I have my training as a biologist, and so I now actually have time. I have discussions with the Ministry of Health and AMA, and it's all very interesting. Everything I wasn't trained for is now actually is actually happening. So interesting.
Mm-hmm. So if you have questions, you can write them in the chat or in the Q&A. There is already a question. Since the normal wild-type allele also has the CAG repeats, how is the allele specificity obtained for an AON?
Yes, that's a very good question. So for some of these polyQ disorders, the allele specificity is actually quite good. For some of them, not as good. So there is slightly more reduction of the wild type protein. And that is also because the structure of these CAG repeats is actually different in some of these diseases. So for Huntington's disease, for instance, we know the CAG stretch is pure CAGs, but for some of these polyQ diseases, there are interruptions in the repeat structure. So we think that the repeat structure is important for the allele specificity, and also the length of the CAG repeat in the wild type allele is important, because for some the wild type length is 17 CAGs, and for other diseases the wild type length can be up to 36 CAG repeats. And then you can already imagine that 36 CAGs is longer, so more of these ASOs can bind to the wild type repeat. So there might be some inhibition of wild type protein expression levels, but we're not quite sure exactly how it works. But we know that the shorter repeats are less likely to be downregulated than the expanded repeats.
Thank you. And then there is a question, what about ASO toxicity?
Yeah, that's also a very good question. So we know that there, especially, there haven't, I'm trying to find a politically correct way of saying this. There haven't been very many successful ASO trials. There have been very many ASO trials for brain disorders, and actually quite a few have stopped due to safety concerns. That's correct. And I think we really need to learn better what the correct dosing is, because the ASOs, they cannot cross the blood-brain barrier, but once they are in the brain, they are active and quite stable for months at a time. And yeah, I think we have been overdosing the brain. You can see it accumulating. And that's also because we don't really know that after intrathecal injections, how much of the ASO is actually ending up in the basal ganglia or in the cerebellum or in the brainstem. And yeah, also there, I think we're still gathering very important information. New studies are being done that will actually probably it will allow us to better determine the correct dose, and I think that will immediately also reduce some of the toxicities that we've been seeing. But we also know there are sequence-specific toxicities that can occur, and especially for the N-of-1 studies, we cannot do primate studies and elaborate PK/PD studies and long-term tox studies just for one patient, especially not if the disease is very progressive. So it's really about finding the appropriate assays in vitro and in vivo to determine the proper tox readouts, and we're not there yet. So I think we really are learning from each and every failed normal clinical study, but also from each and every N-of-1 RNA therapy study that we are doing. And thankfully, everyone is sharing all of their failures and all of their successes. So I think there is really a nice worldwide community that is really dedicated in really trying to move this forward.
Yeah, related to that, Sylvia Bush is suggesting opening the blood-brain barrier.
Yeah, so I think this is one of the options that people have been talking about: intranasal delivery, LMP delivery, a motor reservoir, so you have a direct infusion in the ventricles, I think we've been really, that we can honestly really improve on ASO delivery into the brain. The only caveat is that in Europe, for these N-of-1 developments, we are allowed to sort of have a shorter preclinical trajectory because we use approved ASO delivery methods, and because we use the chemistries that are already approved within FDA or EMA. So there are a lot of novel chemistries also that are probably also less toxic. But EMA has really restricted us on the drugs and the delivery methods that have already been approved within Europe or within FDA. And I don't know of any ASO that has been delivered. I know that there is no ASO that was delivered to the brain with opening the blood-brain barrier in any type of form.
A different question. What about ASO therapy for HD? How close are we for clinical trial stage 3?
So there is a HD generation 2 trial at the moment. Wave has got some trials going on at the moment. I think there are some very interesting small molecules that are being developed at the moment. So I think there is a lot of good stuff happening, and not just on the ASO therapy development. Also there, I think we really dosed very high in Huntington's disease, and I think really, that is probably one of the main drivers of all of why the phase 3 trial was stopped. So yeah, I think we're not there yet. Also not for Huntington's disease.
Thank you. There are no other questions. So thank you very much, Willeke, and thank you for all the participants. And there will be a break of webinars during the summer, and the next webinar will be on the 10th of September. It is related to Huntington's disease. Oh, wait. Sylvia, maybe I can let you speak. Go ahead, Sylvia, you have the floor. You can speak.
Thank you so much.
We don't hear you.
I don't want to speak, I want to hear, and thank you very much.
Oh, okay. She just wanted to say thank you. Oh, there is another question now. Why choosing an intraventricular injection instead of injection through lumbar puncture?
So all of the studies that I did were in mice. So we actually did lumbar puncture in mice, but yeah, the mice is very small. It's actually technically very difficult. Plus, for each injections, the injection volume is so much smaller, and just way less ASO actually travels up to the brain. And so with an ICV injection, you get a higher dose really in the middle of the brain. So you get a very, yeah, it's much more efficient. You don't have to do as many injections. With a lumbar puncture in a mouse, you would have to do multiple injections to reach the same ASO level as with an ICV delivery. And yeah, why we don't do ICV injections in patients, I don't know. I'm a molecular biologist. I don't really know. I think, yeah, these people need long-term treatment. So I think you would have probably the chances of infections are high, and but that would be the ideal. That would be the ideal option. Yeah, for sure. Especially for Huntington's disease, because the ventricles are quite close to the striatum, so you would have a way more direct delivery.
Thank you. Then this is the end of today's webinar. Thank you all, and have a nice rest of the afternoon.